champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to talk about the third topic of biotechnology principles and processes, which is working of the restriction enzymes. It's a very important topic, very fun topic. You'll love studying this. Okay, now over here, what all did we cover in our previous session, children? We spoke about the general idea of genetic engineering, okay, and then we spoke about how is the DNA isolated and how are the fragments of the DNA separated along with the help of their length, okay, and how do we amplify DNA with the help of PCR. So that's the thing that we have completed, and now today we are going to talk about how do we cut the DNA exactly. Before getting into that, let's see the number of questions. So we had 5 questions coming in the previous year from this chapter, which is a huge number because 5 into 4 for all the correct answer. You get 20 marks for this chapter. Something very big, right? Now, I also remember I gave you a homework. Let's see who all got it correct. So during the purification process for recombinant DNA technology, during the purification process, as in when you are separating the DNA, I told the answer, but yeah, okay. So during the purification process, as in when you want to separate things out, okay, and during this process you are putting chilled ethanol. So what does chilled ethanol actually separate out? So if I have a test tube like this and I have something over here and I am putting chilled ethanol, what is it going to separate out? Well, children, when you put chilled ethanol, the DNA, it goes and precipitates. The DNA is that something that goes and precipitates. So your answer becomes DNA, which I already told by chance. This came in 2023. Such an easy question. So if you have studied the previous session, you would be very sure about the answer. It's like this in 1 second, like below that, you might need very less time for answering this question. Now comes today's topic, that is restriction enzyme, starting with the discovery of restriction enzyme. So before going over here, I'll just show you a story of a bacteria and a virus. Okay, so there was a cute little bacteria. Its name was E. coli. Okay, and then there was a virus, children. Okay, there was a virus whose name was bacteriophage. Now, usually what happened is that this particular bacteriophage has its own genetic material, and obviously E. coli also has its own genetic material, which I should not be making in this color. Just give me a second, I'll make it in this color. Okay, it also has a circular DNA over here, and it also has its genetic material, and usually what happens is that this particular bacteriophage comes and attacks this particular E. coli, yeah, and sends its genetic material over here. Okay, sends its genetic material over here, and then this genetic material is later incorporated into the genetic material of the E. coli. So now suppose the genetic material over here, so whenever the DNA duplicates, this also particular portion also duplicates, okay, and whenever anything has to happen, that will happen. So usually what happens, you have the DNA or you have the genetic material, you make what, children? You make mRNA out of it, that is known as transcription, and then later with the help of this mRNA you make the proteins. So since the genetic material over here of the bacteriophage is now incorporated over here, the bacteriophage is now going to use the machinery of E. coli to do the whole process, and it will make its own protein, it will replicate its own genetic material, and now you will get small bacteriophage babies over here, and they will be moving out, my dear children. They will be moving out. So that is the general thing that is happening. But then it was seen that instead of all this thing happening, there was another mechanism that is taking place over here. And what is that mechanism? Let's see. So basically, when the DNA, the genetic material of the bacteriophage is being put over here, okay, the E. coli was producing an enzyme which was cutting this particular DNA strands off into what, children? Into fragments. So they were releasing what, children? The E. coli had certain enzymes which were breaking down the DNA into what? Fragments. Okay, and what were these enzymes? These enzymes were the restriction enzymes. these enzymes were the restriction enzymes which would cut the DNA only at one particular site. So one restriction enzyme will be having one particular site. So if that particular restriction enzymes find that particular site, it will cut over here. It will cut over here, and then it is now in fragmented form. Do you think it will be of any use? No, this particular thing won't be of any use. But on the same hand, the bacteria has to take care of itself as well, because it also might have these particular sites. What do you call these sites as? You call these sites as recognition sites, children. You call these sites as recognition sites. Okay, now these recognition sites which are there, it would be here also somehow. So and if it's here, these enzymes could work here as well and destroy the chromosomal DNA of the E. coli as well. Then what could you do? The thing that could be done is that it can produce another set of enzymes which is known as methylase. And methylase, what does it do? It goes and acts methyl group. It goes and acts methyl group onto the DNA, such that it's a signal that please, it's our own DNA, you should not be doing anything. So these enzymes would not be able to work on these particular strands that you see over here. So what is the use of methylase? It adds methyl group. So as of now, the story goes like this. Usually you had a E. coli and a bacteriophage. This bacteriophage used to come, okay, give out the genetic material. The genetic material used to go and incorporate over there, and using the E. coli's machinery, it used to replicate, it used to make mRNA, the proteins and everything, and then baby viruses would be moving out of the E. coli, destroying it, okay. But this time, the scientists, they saw that okay, the genetic material was still being transferred, but somehow it was getting fragmented within the E. coli's body. And now how did that happen, children? It happened because probably the E. coli might be having certain enzymes which were fragmenting this particular DNA. And what are those enzymes, children? Those enzymes are restriction enzymes. And where are they cutting? They are cutting only at certain recognition site. So one particular restriction enzyme will be having one site itself. So suppose you have a DNA and you have site A, you have site B, and you have site C. The restriction enzyme A will be cutting at site A only. This site as in means there is a specific sequence at which it will be cutting. So for example, a sequence like this AATT, it will be cutting at this particular sequence only. Now how and all, we will get to know that. Okay, so that's how the DNA was getting fragmented. Now if DNA is getting fragmented, or basically genetic material is getting fragmented, if it is getting fragmented, can it do its work? No, it cannot do its work. Going ahead, talking about the enzymes which are there, the next enzyme which is there, okay. So usually you have to protect this particular strand as well. This is the E. coli's own strand, and you have to protect it from these enzymes which they are only making out. So basically, there is another enzyme that is being made, that is methylase. And what does this methylase do? It goes and adds methyl group all over. When the DNA is having methyl group on it, it will be protected. Okay, it will be protected. It won't undergo this particular reaction. You get it? So this was discovered for the first time. This was the thing that was discovered for the first time. So in which year it was discovered? 1963. In 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added methyl groups. What was that? Methylase. And while the other cut the DNA, the latter, the next one, the one that cuts the DNA, was called as restriction endonuclease. Okay, what was it known as? Restriction endonuclease. Endonuclease as in within the DNA if something is being cut. For example, I am cutting from this and here, not at the tips. So when I am cutting in between, you call these enzymes as restriction endo as in within, inside. Nuclease as in nucleic acid. It is cutting the nucleic acid, which is why nuclease term has come. So DNA is a nucleic acid, RNA is a nucleic acid. So here we are cutting the DNA, it is known as restriction endonuclease. If it had to cut outside, it was known as restriction exonuclease. We'll come to talk about that as well. So the first restriction endonuclease was HIND2. What is it, children? Please, you have to remember it. Did I do that? Okay. Its name is HIND2. Well, HIND2 stands for, remember this, Haemophilus influenzae. So you take H from here, you take IN from here, and D is the strain that is there. Okay, so you have different strains of bacteria. D stands for that particular strain, and 2 as in this was the number second enzyme that was being, I don't know why they put it number second. Okay, anyways, I'm not making you confused. This basically denotes the sequence in which the enzymes were discovered. Okay, so anyway, hint 1 is not mentioned, hint 2 is mentioned. You have to remember hint 2 because that is the one that was being actually discovered, and I don't know why they named it like that. Anyways, now whose function depended on a specific DNA nucleotide sequence was isolated and characterized 5 years later. So it was later on isolated and found out. Okay, but you have to remember the name. It is hint 2, children. What is it? Hint 2. Now it was found that hint 2 always cut the molecules, DNA molecules, at a particular point by recognizing a specific sequence of 6 base pairs. So this hint 2 was always cutting a specific sequence which had what, children? 6 base pairs. Okay, no other sequence. Always the sequence was same. I'm not writing the sequence over here because it is not given in your NCERT. Now this specific base sequence is known as recognition sequence for hint 2. Now this is very specific about having this cutting in this particular sequence. So what do I call this? I call this as recognition site or recognition sequence. Fine. Now besides hint 2, today we know more than 900 restriction enzymes. There are 900 restriction enzymes, each of them having their own recognition sequence as well. And these 900 restriction enzymes were discovered or were isolated from 230 strains. How many strains, children? 230 strains. So from 230 strains, you got 900 restriction enzymes, each of them having their own recognition sequence. So from this particular slide, you got to know that it's 1963 where first time it was observed that something like this is happening. Okay. Then later on, and what were the enzyme? Methylase. One was adding methyl group, it was methylase. Second one was cutting the DNA, it was known as restriction endonuclease. Now for the first time, the thing which was isolated, the first restriction endonuclease which was isolated was hint 2. Okay, it was hint 2, not 3. Please do not get confused over here. I actually did that again. Okay, anyways, so that is the thing. Now going ahead, what do we have? Naming of the restriction enzymes. How do you name it? I have already told you, but proper way of naming it, I'll tell you again. So the convention for naming these enzymes is the first letter of the name comes from the genus. So for example, you have one enzyme that is known as EcoR1. Here E stands for Escherichia, okay, and CO stands for coli. Over here, this thing that you see is the genus name and this is the species name. Okay, so over here I take E, then I take CO, the first and the second letter from the species name. Okay, the first and second letter of the species name from here, and this stands for the strain which is there. So here R stands for RY strain. There is a strain which is known as RY strain. And 1 stands for what, children? I will tell you about that. So Roman numbers following the names indicates the order in which the enzymes were isolated from that strain of bacteria. So this is the order by which the strains were actually, so the enzymes were actually taken out or isolated. So this was the first strain to be taken out. Okay, so for hint 1 and hint 2, please do not get confused. It's hint 2 that was isolated. I don't know why there is hint 1 and that not been mentioned. There might have been something. Okay, anyways, so EcoR1 over here, E will stand for the first letter of the genus name. The next 2 letters are from the species name. The first 2 letters of the species name. This one will be standing for the strain. Here it is RY, and 1 is basically the order in which it was discovered. Fine, cool, works. And just give me a second. Okay. So the convention for naming these enzymes, the first letter comes from the genus and the second 2 letters comes from the species of the prokaryotic cell from which they were isolated. Example, EcoR1 comes from Escherichia coli RY13. Now this is very much important. So RY13 is the strain. Okay, RY13 is the whole name of the strain. Now in EcoR1, the letter R is derived from the name of the strain. I told you, name of the strain is RY30. Roman numbers following it indicates in which order the enzymes were actually isolated from the strain of bacteria. Restriction enzymes belong to a larger class of enzymes called nucleases. I told you. Basically, what do you mean by nucleases? Nucleases are the ones that acts on nucleic acid. What about proteases? Proteases are the one that acts on proteins. What about lipases? It's the one that acts on lipid. So when the ACE term comes, just understand you have to look for the term before it. So they act on nucleic acids, and what all nucleic acid do we have? We have DNA and we have RNA, my dear children. Deoxyribonucleic acid and ribonucleic acid. Now over here in the case of nucleases also you have two different types. I told you one is exonucleases. It's the same term, okay? And the other one is endonucleases, okay? So exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases makes the cut a specific position within the DNA. So exonucleases, if for example this is the DNA strand, they will be cutting towards the end. That is a function of exonucleases. Talking about endonucleases, they will be cutting, for example this is the DNA strand, they will be cutting in the middle regions like this. Fine. Now that's also done. Next thing, the working of the restriction enzymes. How do they actually work? So we have to see that now. Chalo. So before reading this, we have to study it, and it goes like this. Suppose over here, suppose we have to cut the DNA strand, okay? And I am saying I am going to use a very important type of enzyme for that, that is EcoR1, okay? EcoR1. And what is the sequence of EcoR1? I told that all the enzymes has a recognition sequence or a recognition site. What is the sequence for EcoR1? It is 5' GAATTC, and it will end up in 3'. This is the recognition sequence, okay? Now I am telling you the recognition sequence, no, they will be always palindromic sequences. Either way if you read, they should be same. So GAATC is over here, it's okay. Now I am saying this is also its recognition site, where it is CTTAAG. So if you read, just a second. If you read from this to this end, it is GAATTC. Again from this to this end, it is again GAATTC. So recognition sites, no, it will be always known as palindromic sequences. They will be always known as palindromic sequences. What do you mean by that? When they are read in both the directions, it's the same thing. So Malayalam, if we take, it's a word. Malayalam, all Malayalis assemble over here. Malayalam, right? So if you read from here, if you read from there, it's the same thing. Similarly over here also, if you read from this side, if you read from this side, it's the same thing. But this is a sequence. It has to go in 5' to 3' direction. Again, it has to go in 5' to 3' direction. So this is the recognition site for EcoR1, and you have to remember it. Questions have come from this. You have to remember the site for EcoR1. Now, I am going to cut the DNA in this particular format. Okay, I want to cut the DNA. So for that, what I do? Basically, suppose this is the DNA strand, okay, and this is the 5' end, this is the 3' end. This has to be 3', this has to be 5' because they are anti-parallel in nature, okay. Now over here, I suppose it's GAATTC, okay. Then here what it will be? It will be CTTAAG, okay. Again over here there will be some other sequences at the end. At the end again it will be GAATTC over here, and this will be CTTAAG over here, okay. And here you have other DNA sequence strands. So we want this particular DNA sequence strand. So here also you have so much amount of DNA, there also you will be having so much amount of DNA anyways, okay. Now I said that this is the what, children? This thing that you see over here, this is the recognition site. This is the recognition site, and the enzyme is able to find it after searching for it for a long time. Okay, it goes through the entire sequence and then it finds that, okay, here the sequence is there, I have to cut it over here. Okay, that is the function of the enzyme. Now, it does not make a cut at middle. It usually does not make a cut at middle, the enzyme that you see over here. Rather, it will cut in between G and A. So if it is cutting here in between G and A, that means here also it is going to cut between G and A. Okay. So the site, the actual in the recognition site, the area where will actually cut will be in between the G and the A. It will cut in between G and A. So this is the palindromic sequence. I'll say it is not cutting in the center of the palindromic sequence, but is cutting a little away from the center. What do I say? It is cutting little away from the center. All these details that I am saying is very much important. Okay. Now, if I am saying the cut has to take place over here with the help of this enzyme. So this enzyme has come over here to cut the DNA. Let's see how does it look. So it found the sequence, it cut it over here. It found the sequence and it cut it over here. It found the sequence and cut it over here. Okay. Similarly, it found the sequence, cut it over here. It found the sequence and cut it over here. Now what will you be left with? That is something that we have to make. So let's see. So here, when I cut this off, what do I get? I have A, T, C. Agree? Over here, I just have G at the end. Now, talking about this, I just have G over here. Rest is the DNA. But over here, I have C, T, A. This is the 5' end. This is the 3' end. Similarly, this is the 5' end and this is the 3' end. So over here you see this C and G have complementary base pairs, so they pair. So they are still together. But what about them? Rest all the DNA strand, it's still together, okay? This strand is still being together. Just the portion that I have cut, okay? The portion where I have cut, that is until here I have cut over here, but over here I have cut until here. So anyways, I'll get something like this over here and something like this over here. This does not have any complementary base pairs, so I call this as a sticky end. What do I call this as, children? I will call this as a sticky end. Over there also, if I see, it also has no complementary base pairs over here. Again, can I call it a sticky end? Yes, I can call it a sticky end. I hope you're getting. So we cut it from here, these things went off. These things are not needed, it went off. It's there in the DNA strand, okay? I took this particular DNA strand now over here, okay? This is what I have got. This is my gene of interest. This is what, my children, this is the gene of interest. Now, what am I going to do with the gene of interest? Let's see that. So, I took this with the sticky ends and now I have, okay, now I have a plasmid. Now I have a plasmid, and here in the plasmid also I will be using the same enzyme, that is ECO R1. I will be using the same enzyme, that is ECO R1, and I will be cutting the strands over here as well. So if this particular thing has a recognition site for ECO R1, it will cut at the same site. It will cut at the same site. That means here also I will be getting sticky ends. Here also, children, I will be getting sticky ends. So it will look like this. There is this strand, okay, and there is this particular strand, something like this. Now, if I take the DNA that I have over there, what is the DNA that looks like over there? It is like this. So it has TTC over here, and then there is G over here. Then again over here also it has AA. Just a second. I have to extend a bit. Okay. Over here also it has AA, T, C. There is just G over here. So you had sticky end over here, you had sticky end over here. This thing, if I go and pair over there, because this also has the same thing, this will also have the same sequence. What is it, children? It will be having the complementary sequence AA. What is it? AATT, right? AATT. It will be having AATT over here. the complementary one over here. Similar, because it is also cut in the same way. It is also being cut in the same way. Suppose it was G over here, but I make it cut over here. So now what do I have? I cut the G over here. I have a strand like this remaining. Okay, this is the sticky end that is being remaining over here. Similarly over here also, if I see, it will also have something like this. Here also, it also will be having something like this, that is AATT. Agree? It will also have something like this over here. I can write AATT. Right? Now this strand will come and bind over here. If I put my DNA strand totally, what do I get, children? Basically, they will be sticking onto each other because both of them will be having the same sticky ends. Both of them will be having the same sticky ends because they are also cut in the same way. So they will be having a complementary base pair over here. That is, the bacteria will be, the chromosome will be having a complementary base pair over here. Similarly, here also, it will be having a complementary base pair. Here also, it will be having a complementary base pair. Both of them will now stick to each other. Okay, both of them will now come and join because this is complementary, so obviously they will join. Now there is one more thing. The joining has not yet completed. Joining has not yet completed. It will be only completed when I put DNA ligase. When I put DNA ligase, that's the time when the joining will be officially completed. So DNA ligase is the one that joins through different type of DNA over here. Okay, so this is what the plasmid DNA that we had from E. coli. Okay, this is, suppose this was, I was putting my gene of interest in E. coli's plasmid. So this is the plasmid that I got from E. coli. Now I am putting my gene of interest over here. Both of them have to come together, even though they have bonded with the help of complementary base pairs because they were cut by the same enzyme. They still have to come together with the help of one enzyme which is known as DNA ligase. Okay, did you get it? This is how things are going to work. So these sticky ends are kind of very much important. These sticky ends that you see are very much important. If sticky ends are not there, okay, if it is not cut like this, then obviously you do not have a space for including some other, DNA. Okay, so that is why sticky ends are very much important. Now we will read and see if you are able to understand things or not. Chalo. First point, each restriction endonuclease functions by inspecting the length of DNA sequence. What children? It inspects the length of DNA sequence and then only it works. This is a very important point. You have to learn it. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific points in their sugar phosphate backbone. So they are going to make a cut where, children? In the sugar phosphate backbone. So they are going to cut the phosphodiester bond over there. That is how it works. Okay. Now each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. So all of them will be having a palindromic sequence. For EcoR1 we saw it was GAATTC. This is 5' to 3' dash, and again 5' to 3' it will be same thing, GAATTC. Okay. Restriction enzymes cut the strand of DNA a little away from the center. So they are not usually cutting in the center, they will be cutting it away from the center. So there are certain restriction enzymes which does cut in between, but we are not going to study about that. Okay, but between the same two bases of the opposite strand. So okay, so they are saying that it is being cut, okay, in the palindromic sequence away from the center of the palindromic sequence, and they are cut between the same two nucleotide bases. So this is guanine and adenine, here also it is guanine and adenine. So palindromic bases, but between the same two bases on the opposite strand. This leaves single strand portions at the end. It will be leaving single stranded portions at the end. Okay, so over here if I cut, it will be AATTC. What will I get over here? Here I will get nothing but G, because I have cut this portion, I have cut this portion. Got it? Now, okay. So I am getting a sticky end. That's the thing that I want to tell. So these are the overhanging structures which are known as sticky ends on each strand. These are named so because they form hydrogen bonds with their complementary cut counterparts. this stickiness of the ends facilitates the action of the enzyme DNA ligase. So if the stickiness was not there, DNA ligase probably might not have worked properly, right? So it's DNA ligase's function is to just join the DNA. That has to be done properly. So when cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and these can be joined together end to end using DNA ligases. So since it is cut by the same restriction enzymes, both the plasmid and the gene of interest, it will be able to ligate with each other very easily, and here you will be needing the help of DNA ligases. Fine. So that is there. Now one more thing is given in your NCERT, that is what are palindromic sequences. So do you know what are palindromes? These are groups of letters that form the same words when read both forward and backward. Example, Malayalam. As against a word palindrome where the same word is read in both directions, as you can see over here. Okay, so for example, the following sequences reads the same on the two strands in 5' to 3' direction. So 5' to 3' direction, it is GAATTC. Here also it is GAATTC. This is the sequence of what, children? Eco R1. So usually they do not ask the sequences of other things, but sometimes they could ask. Okay, once they have asked like that, but it's kind of very rare. So that is all about it. Fine. Now, I'll just repeat what I just taught. So in 1963, what did we have, children? The first, for the first time it was seen that in E. coli there was something happening. There was some enzymes which are cutting the DNA. There were some enzymes which are adding methyl group, that was known as methylase. Then later on, the first restriction enzyme was actually isolated, and that was HIND2. It is Haemophilus influenza D strain, okay, and it was second strain over there. Now going ahead, we spoke about how was the naming done. So the first letter of the enzyme comes from the genus's first letter, okay. The second two letters, okay, the second letter and the third letter comes from the species name, okay. The first two letters of the species name becomes the thing over there. And now R term came from the strain, okay, it was RY strain, RY13 strain of E. coli, which is why it was R. The name R came. And now the numericals which follows, for example, Eco R1, 1 comes from in the order of which the enzymes were actually isolated. Cool. After that, we spoke about the restriction enzymes and their working over there. We studied about Eco R1 site. The site is, recognition site is GAATTC. What is it, children? It is GAATTC. Over here, they make a cut between G and A nucleotide. Okay, whenever they make a cut, they are going to give rise to what? Sticky ends, and these sticky ends are very much important for what, children? For the genetic engineering to be done properly. Now if sticky ends and everything are there, they are complementary to each other, they will go and stick for sure, but they still need the help of an enzyme which is known as DNA ligase. Khatam. That's all. Okay, now identify the wrong statement over here with regard to restriction enzymes. That is the first question. I will just read the question. Later on, I will be solving it. It's the time for you to solve now. So each restriction enzyme functions by inspecting the length of a DNA sequence. Okay, does it inspect the length of the DNA sequence? Okay, they cut the strand of DNA at palindromic sites. Do they cut at the palindromic sites? They are useful in genetic engineering. Are they useful in genetic engineering? Sticky ends can be joined using DNA ligases. Tell me, can they be joined using DNA ligases? You have to tell me. Okay, NEET 2020 question is that. Now next question, second question. The specific palindromic sequence which is recognized by Eco R1 is what? So you have to go through each and every one and tell me the correct sequence. Okay, you can pause the video and try it for yourself and then only tell me. Next one, third question. Okay, you can answer like, ma'am, first question answer is this, second question answer is this, and then go on. So the following statements describe the characteristics of the enzyme restriction endonuclease. Identify the incorrect statement. Okay, the wrong statement you have to tell. The enzyme recognizes a specific palindromic nucleotide sequence in the DNA. Does it? The enzyme cut the DNA molecule at identified position within the DNA. The enzyme binds DNA at specific sites and cuts only one of the two strands. The enzyme cuts the sugar phosphate backbone at the specific sites on the each strand. This came in NEET 2019. Again, if you have not read the NCERT, will you be able to answer that? It's not just about reading the NCERT. If you have not paid attention to what was important, will you be able to answer this? Obviously no. which is why I said all the points are kind of very, very, very important. You never expect some questions to pop out just like that, but it comes. Okay. So, question number 4. A foreign DNA and plasmid cut by the same restriction endonuclease can be joined to form a recombinant plasmid using what? EcoR1, Taq polymerase, polymerase 3 and ligase. So, foreign DNA and plasmid that are cut by the same restriction endonucleases, they can be joined, they can be used to make a recombinant plasmid. Okay, it is done with the help of what? EcoR1, Taq polymerase, polymerase 3 or ligase. One very important thing. So, now when the plasmid has the gene of interest, you call this as a recombinant plasmid. You call this as a recombinant plasmid. Now, when you are putting this recombinant plasmid into a bacteria, suppose it's E. coli, you will say that this is transformed. This particular organism that you see over here, it is transformed. That is also of very much importance. Cool. That's the 4th question. Now, the 5th question that we have. Which of the following is a restriction endonuclease? You have DNase 1, you have RNase, Hind3 and you have Hind2 and protease. Which one is a restriction endonuclease? This is a very easy question. This came in NEET 2016. If you see that type of question is kind of very easy, just that you should be reading a bit of NCERT. That is the most important thing over here. Okay. So, I hope you got the questions, you answered it as well. Now, please tally your answers with me. Okay. So, the first question says, identify the wrong statement with regard to restriction enzymes. Each restriction enzyme functions by inspecting the length of the DNA sequence. Yes, it does. It does inspect the length of the DNA sequence, and this is what I said. Now, they cut the strand of DNA at palindromic sites. Yes, they definitely cut at palindromic sites. They are useful for in genetic engineering, my dear children. Are they useful for genetic engineering? Yes, they are definitely useful. Sticky ends can be joined by using DNA ligases. DNA ligases are not for joining sticky ends. Basically, it's for making 2 DNAs come together. Okay, for making, this is the plasmid DNA and you want your gene of interest DNA to come together, to ligate together. That is why you use DNA ligases. So, this becomes the wrong answer. This is the wrong statement. Okay, works. Now, the specific palindromic sequence which is recognized by EcoR1 is GAATTC, right, and it will be in 5' to 3' direction. So, this has GAATTC in 5' to 3' direction. Similarly, GAATTT again in 5' to 3'. Please do not go with this. Here the sequence is wrong, here the sequence is wrong, here also the sequence is wrong because it is 3' to 5' direction, but it has to be 5 to 3. So, this becomes correct. Cool. The foreign DNA and the plasmid cut by the same restriction endonuclease can be joined. The foreign DNA, that is this, and the plasmid, okay, cut by the same restriction endonuclease can be joined to form recombinant plasmid using what, children? Using the ligase. Here the DNA, this particular DNA is made to join with the help of ligase. You know that is the enzyme for joining. Now, following statements describe the characteristics of the enzyme restriction endonuclease. Identify the incorrect statement. So, enzyme recognizes a specific palindromic nucleotide sequence in the DNA. It is correct. The enzyme cut the DNA molecule at identified position within the DNA. Yes, exactly. It is correct. It will identify, then only cut it, right? Now, the enzyme binds DNA at specific sites and cuts only one of the 2 strands. It is wrong. It will cut both the strands. It never cuts one strand. It cuts both the strand. So, that is wrong. That is the incorrect statement. This is the answer. The enzyme cuts the sugar phosphate backbone. That is also correct. Now, fifth one, which of the following is a restriction endonuclease? The one that cuts the DNA. Sorry, that is cutting the DNA palindromic sequences in one particular site. What is that? Restriction DNase 1, RNase, protease, Hind2. This will be Hind2. That this was the first one that was isolated as well. Works? Okay. Chalo, let's move on to the next question. Oh, we do not have next question. We have 5 questions and I hope you're done with that. I have one little homework for you as well, and over there just wait for a second, okay? Yeah, so that's all. And one more thing I want to say in this question, no, I know this is kind of very confusing, okay, which is why even I had to go and have a look. So this is the correct answer for sure, but then yes, I want to say why they actually said that this is a wrong option is that sticky ends, they suppose that hydrogen bonds are enough for them to join from to each other. So you have suppose where is it, suppose you have AA over here, then you have TT over here. Obviously they will make hydrogen bonds easily, which is why the sticky ends, it is written that sticky ends necessarily do not need DNA ligase to join itself. It's just that the DNA fragments, the gene of interest and the plasmid DNA, if you want to pakka seal them, for that you need DNA ligase. So I just wanted to give a brief explanation of it. I don't know how far you also have thought it is correct, because this is actually a vague question. They should not be putting questions like this, but yeah, it has come and we have to choose the most appropriate one. So other options were totally fine. This was the only thing that was a bit awkward, so that's why we went for that. Okay, now we have got a little homework for you cuties. So given below is a sample of a portion of DNA strand giving the base sequence on the opposite strands. What is so special shown in it? This is a strand, the opposite strand given over here. Does it show replication is completed, deletion, mutation, start codon at 5' end, is the start codon at 5' end, palindromic sequence of the base pairs? What is so very different for it? Now this is again a very easy question, I hope you can do that, but I wanted to put it over here because for someone who has studied molecular biology and everything, you can think this is also true. You can think this is also true, but again you have something more specific over there, then you should be going for that. This is which is why I gave this as a question. Okay, I partially gave the answer as well, but still. So that's all, my dear little champions. I just want you to study this and do let me know if you have any confusions. I'll surely clear it up. Okay, so that's all. Please take care of yourself and have a very good health and be happy. Everything is going to be all right. Bye bye.